Thank you so much, Stacy, and to the AAPS, and I hope you can hear me okay. It's really the pleasure to be here today, and thank you, of course, for the warm introduction. As Stacy mentioned, my name is Jain, and I'm here on behalf of Sapient today, and I'm really excited to chat with everyone about Sapient's efforts to leverage circulating biomarkers to make clinical trials smaller, faster, and more effective. Next slide, please. So to understand how we can make clinical trials better, it is really important for us to understand how clinical trials go awry. And I think this is illustrated perfectly in a recent review and a recent analysis of 85 FDA-approved cancer therapies, which are summarized here on the right. And what we find is that if we look at the median response rate for each of these therapies in their individual clinical trials, it's only 41%. Of the 85 approved agents, 14 or 16% of them had a response rate less than 20%, and 28 drugs, or about a third of them, had a response rate of less than 30%. Only a handful of the therapies actually work on more than 50% of the individuals in an individual clinical trial. And herein lies the fundamental challenge, and that is that the majority of patients enrolled in any given clinical study don't actually respond to the actual therapy. And again, to reiterate, these data are for FDA-approved therapies. Next slide, please. So why is that? Well, diseases are really defined by a singular pathology, but in fact, most diseases represent a grouping of contributing factors and pathways that produce a singular shared pathologies. So in other words, diseases may, for all intents and purposes, look indistinguishable on pathologic examination, but may have very different pathophysiologies that lead them to go from an individual going from a normal state to a disease state. And as drugs largely target underlying mechanisms of disease, it would be expected thereafter that only a portion of individuals would respond to any given therapy. Next slide, please. If you don't mind advancing one more slide. There we go. Another way to evaluate the relative efficacy of drugs is through number needed to treat analysis, which indicates the number of individuals that must be treated for one individual to benefit, as defined by the trial outcome and over the trial duration. To give you an example, a number needed to treat of 1 would indicate that everyone benefits from the therapy, and the higher the NNT, the less likely the drug is to benefit any singular individual. Now, an NNT of 5 is considered pretty good by pharma standards, but that means that of the 5 people tested, only one of them is really actually having a true benefit with regards to the underlying outcome, which means that the other 4 individuals didn't benefit while absorbing much of the potential harm for any particular therapy. Now, if we look at common therapies, we find just how broad the spectrum from a number needed to treat perspective can actually be. Triple antibiotic therapy for H. pylori has an NNT of 1.1, meaning that virtually everyone that receives this therapy benefits. Sumatriptan for migraines has an NNT of 2 to 3, and so on and so forth. Now, the NNT particularly begins to fall off significantly when we think about prevention of disease. And we see this illustrated with statin therapy, which for secondary prevention of myocardial infarction in those with established coronary artery disease, the NNT is approximately 15. Whereas for primary prevention, as shown in the last row here, in those without established CAD, statin therapy has an NNT that's greater than 100. Which means for every one individual that has a prevented heart attack because they took a statin therapy, 99 individuals at least did not actually benefit. Next slide, please. In addition to the risk to the actual patient, there's significant cost in terms of expense and time to those engaged in drug development. We know that drug trial costs do not scale linearly, and a 10x increase in trial size is often associated with a 12 to 15x increase in cost, which can easily scale into the hundreds of millions in today's world, as shown here on the left. Equally as expensive is the time. Larger clinical trials require longer timelines to recruit, enroll, and monitor patients. And even then, after this time and expense, only a fraction of drugs will advance to actual approval. Next slide, please. Ultimately, even for those drugs that do meet FDA approval, it largely remains a coin flip at best as to whether any given individual will benefit from the treatment. For those that don't benefit, again, they're absorbing all the potential risks of taking that drug, as well as, importantly, not receiving alternative treatments which may specifically benefit them. Next slide, please. Now, this current state of drug trials and drug development has prompted much interest in strategies aimed at clinical trial enrichment, to improve this fundamental issue, and approaches specifically for tailoring therapies in a much more precise manner, particularly based on clinical or broad-scale omic biomarkers. These strategies have been captured in a recent white paper for the FDA on clinical trial enrichment strategies, which outlines approaches for using biomarkers to enrich clinical trials in a much, much more targeted manner. Next slide, please. Now, essentially, what biomarkers allow us to do is they enable us to stratify and enrich patient populations, fundamentally allowing one to align the specific patient with a specific disease state and a specific therapy, whether it be for a small molecule, large molecule, gene or cell therapy. In this way, one can theoretically identify those patients that have a specific pathobiology or a mechanism that allows them to go from a normal to a disease state, and specifically treat these individuals with therapies that target that specific pathobiology in a manner that greatly increases the underlying response rate. Next slide, please. Now, there's actual hard data to suggest that this is more than theoretical. An independent retrospective analysis now across over 20,000 clinical trials from the last several decades have revealed that those drugs that are developed together with a biomarker had a 2 to 10-fold increase in FDA approval. This spans multiple areas from oncology to cardiovascular metabolism and metabolic disease, infectious disease, autoimmune disease, and et cetera, et cetera. Not only is the approval actually higher, but the time to approval is quicker. So not only are biomarkers associated with more effective, but actual faster clinical studies. Next slide, please. Now, you can actually begin to model this to determine just how much smaller and more effective clinical trials can get through the use of biomarkers. This table is taken from the FDA white paper that I just mentioned several slides earlier, and illustrates how sample size ratios, theoretically defined as the prevalence of biomarker-positive individuals in any particular study. As the prevalence of these individuals in a study decreases, the sample size has to increase. As shown in the first row, if everyone in a clinical trial is biomarker positive, meaning they all have the same disease and the same pathobiology and are likely to have the same response to a particular therapy, clinical trials can be quite small. But as the prevalence of a biomarker positive patient drops below 15%, particularly as we go down in this table, clinical trials must increase exponentially in size. And you can see as the percentage of individuals that are thought to have a biomarker or respond drops below 25%, a clinical study must be over 16x the size with regards to their underlying enrollment. Next slide. Now, this assumes that biomarker negative patients have no response. And even if they have a 50% response rate by current standard therapy, you can see as the biomarker positivity drops, clinical trials still must be quite a bit larger, though this increase now is linear as opposed to exponential. Next slide, please. So we can begin to see how this translates to actual real-world clinical data. Shown here is data from the phase 3 clinical study of trastuzumab, which targets, of course, the HER2/neu receptor, which is overexpressed somewhere on the order of about 25% of breast cancer patients. When compared to standard of care, a non-enriched clinical trial on the left bar graph here of all comers required thousands of individuals to demonstrate overall efficacy, particularly as this efficacy is limited to biomarker-positive patients who had a 10% improvement in one-year disease-free survival. Now, using an enrichment strategy here, in this case, HER2/neu positivity as a biomarker, a clinical study could be reduced by overall approximately 16x in size while still maintaining statistical power for detection of the overall benefit. And you can see this on the right bar graph here. You can see that the effect size or the proportion of responders on all comers on the left and the blue and red bar there. And then in an enriched clinical study, again, one that is about 15 to 16-fold smaller in size, the statistical power for detecting a positive response is preserved or even enhanced among control and treatment individuals. You can imagine this type of enrichment has massive implications for clinical trial design. Next slide, please. Now, there are many different types of enrichment markers that may be leveraged for clinical trials, including diagnostic markers that identify a subset of individuals with similar disease pathobiology. There's predictive biomarkers that identify patients most likely to respond to a given drug therapy. There's prognostic markers that identify patients according to progression rates and likelihood of developing clinical events. Alternatively, there's safety markers here on the bottom that identify those that are most likely to have an adverse event, and even monitoring markers or surrogate endpoints that provide information regarding clinical trial benefit to a particular patient, even at a very early disease point. Please a bit. Now, to date, much of the clinical biomarkers that have been employed have really centered on genomic markers. And while these have had profound influence for enrichment among cancer patients, it's clear that underlying genetic predisposition only accounts for a portion of risk of developing most common diseases. If we look at this analysis shown here on the right across hundreds to thousands of mono and dizygotic twin pairs, we can begin to estimate the fraction of population attributable risk that's due to genetic versus non-genetic risk factors. And perhaps not surprisingly, for most human diseases, whether it be cardiovascular disease, neurologic disorders, pulmonary, autoimmune, or other diseases, genetics only account for a minority fraction of risk. And upwards of 80% of all population attributable risk of disease is still unaccounted for, I should say, by genetic factors, and therefore largely remain unexplored. And here's where small molecule biomarkers hold enormous values. Small molecule biomarkers read out non-genetic factors underlying human disease. They reflect disease-specific processes. And given their relatively small mass and underlying chemistry, these can easily be released across cellular, tissue, and biological membranes from a particular peripheral tissue bed into the central blood, and can therefore reveal tissue-specific pathology and signaling at a very early time point from simple blood samples. Next slide. Now, here at Sapiens, we've built a biomarker discovery engine, and we really view ourselves as a discovery partner that functions through three integrated pillars to accelerate biomarker discovery. The first pillar on the left here is an underlying next-generation proprietary mass spectrometry technology that allows us to capture and assay more than 11,000 small molecule biomarkers across a host of biosamples, from blood to urine to CSF to tumor to tissues to even cells in a dish at remarkable rates of speed at under 1 minute analytical cycle time per sample. This system is particularly geared to the non-targeted measure of small molecule biomarkers across a range of chemistries, from very polar to very nonpolar molecules, including thousands of yet unmapped and yet undiscovered small molecule biomarkers. The second component, shown here in the middle, is a deep expertise in biocomputational learning that allows the Sapient team to apply machine learning and AI-based algorithms to integrate multidimensional data, including thousands of small molecule biomarkers, with preclinical data, canonical markers, cytokines, genomics data, biological factors, and most importantly, clinical outcomes to take complex data and derive actionable singular insights. And the final component that's shown here on the right is a rich human biological database of hundreds of thousands of clinical biosamples, which have already been analyzed using our proprietary mass spectrometry systems. And these clinical samples include those with decades-long longitudinal clinical outcomes data, demographic information, drug response, complex genetics, microbiome data, as well as lifestyle factors. Next slide, please. Now, these three components coalesce to enable Sapiens discovery of small molecule biomarkers at scale. If you could just please advance there, Stacy. So what are some of the real-world applications for these small molecule biomarkers, particularly within the context of clinical trials? Well, we've put together here several examples by which these approaches have been used. The first is a discovery made together with a Sapient partner to identify new pharmacodynamic markers that may reveal drug responders. Some of the details, as you can imagine, I'm unable to share given confidentiality of the data, but hopefully these serve as conceptual overviews that will allow you to understand the value of the underlying approach. If you just don't mind advancing there. Cool. If you could advance one more, please. Now, this work leverages biosamples from 250 or so individuals enrolled in phase 3 clinical trial that failed to meet its primary endpoint due to a lack of efficacy. In each of these biological samples from before treatment and after treatment, we were able to capture their blood samples and assay using our proprietary technologies 11 to 15,000 small molecule markers in each of these biosamples, which is shown by these little gray dots on the volcano plot here to the left. Now, using this complex data, we can simply ask, are there markers that are changing in blood in response to a treatment? And in doing so, have identified a number of potential pharmacodynamic markers which are altered in response to drug exposure. From this data, biocomputational analysis prioritizes one such small molecule biomarker, which is circled here in the left panel in blue. As shown on the upper right, this biomarker is particularly variable and altered in a subset of individuals following treatment, but remains flat in other folks who receive therapy, as you can see from the baseline to the post-treatment lines here. Now, what makes this biomarker particularly compelling is that if we go back to the initial clinical trial data, and again, this was a drug that failed for its primary mortality endpoint, and we subset the data according to those that had low levels versus high level of the marker post-treatment. We find that those individuals in which the the marker was elevated are actually quite responsive to the therapy, where those that had flat levels after treatment continue to have a progressive decline. And theoretically, this pharmacodynamic marker serves as an excellent approach to subset individuals into responders versus non-responders. Next slide, please. Now, this particular biomarker is an unknown small molecule marker that we were able to isolate and solve its underlying structure. And across Sapiens human biological databases that I previously described, there's much we can learn about this marker and its related markers in blood. Across over 100,000 individuals in our underlying database, we find that this marker, as well as related markers, are very closely aligned to underlying tissue biology. And individuals with higher levels of the related marker are associated with more rapid disease progression, as shown on this Kaplan-Meier plot on the left. In tens of thousands of individuals, we have underlying genetics data and can perform genome-wide association to identify those common genetic variants that alter the level of the marker in circulation, as shown here on the bottom right Manhattan plot. And doing so, we can reveal the key biosynthetic pathways that give rise to the marker, its transport mechanisms, and given where those genes are actually expressed, even the tissue or cell type from which the marker originates. And in doing so, can learn quite a bit about the underlying biology of a specific individual marker. Next slide, please. Now, similarly, we can identify prognostic markers that also enrich for clinical events, which is critical in thinking about how to enroll folks in clinical trials where there's a desired outcome event rate. If you don't mind advancing, please. Now, across clinical trials, we often use surrogate markers of disease to enrich for individuals that are likely to have a disease-related event over a given period of a clinical study. Selecting individuals at high risk, of course, is balanced across the need to screen more and more individuals to find those individuals that have high risk. For instance, in the setting of renal disease, these markers may be GFR or proteinuria, or in heart failure, the presence of low cardiac function. Herein, we can use a broad measure of thousands of small molecule markers across multiple independent studies, totaling tens of thousands of individuals, to identify machine learning-based biomarker risk scores that identify individuals at high risk for events compared to standard enrichment strategies. Shown here on the left, when compared to standard of care, we find that a derived biomarker risk score identifies individuals at high risk for clinical events across multiple independent studies. And shown here on the right, when we compare this to standard enrollment strategies, we find that this biomarker risk score at varying thresholds, as shown as the orange dots, enriches not only for events, allowing for less individuals to be enrolled, and given its ability to detect those that are at highest risk for events, even requires less individuals, or fewer individuals, I should say, to be screened. Again, you can imagine how this approach may greatly enrich clinical study enrollment and allow for studies that enroll faster, as well as ultimately can be smaller in size. Next slide, please. Now, across our biological database, we can similarly determine that this biomarker, as shown in the last example, selects individuals that are at highest risk for rapid progression of disease over time, validating its potential as a clinical enrichment strategy. If you don't mind advancing. Now, in the same manner that we search for diagnostic or prognostic markers, we can similarly identify biomarkers of safety that allow for subsetting of patients that are at high risk for development of an adverse drug event. Together with a number of partners, we have engaged in these efforts to identify circulating markers that denote individuals at high risk for severe adverse immune-related events in the setting of solid tumor immunotherapy. These severe autoimmune reactions can be quite debilitating, as you're quite aware, and limit overall dosing and efficacy of these therapies. If you don't mind advancing. Now, across multiple independent clinical studies of solid tumor immunotherapy, in total encompassing over 300 individuals, we can perform a broad biomarker discovery, again measuring thousands of circulating small molecule biomarkers, as represented by the small gray dots on this left panel volcano plot. When we examine these markers in relation to development of severe immune-related adverse events, we find that one marker in particular is prioritized, again shown here as the blue circle. In independent data sets in the upper right panel, we find that across patients with solid tumors that undergo immunotherapy but don't develop autoimmune adverse events, the marker is relatively stable as time, as you can see on the left part of that panel. Similarly, in those individuals that develop mild IRAEs, such as mild colitis, diarrhea, etc., the molecule is stable over time. But as shown on the upper right, in the right panel, in those individuals that develop severe immune-related adverse events, this marker in particular drops quite precipitously. And really importantly, this drop precedes the clinical manifestations of autoimmunity by several weeks, providing now a potential window for secondary therapies to be instituted. Now, this is a marker that we've confirmed in preclinical models as well. On the bottom right is a humanized CTLA-4 mouse model, when exposed to anti-CTLA-4 therapies, develop a rather severe autoimmune colitis. And as you can see, those animals that have lower levels of the marker develop much greater autoimmune-related toxicity. Now, this is a marker also that we have independently validated and confirmed in a number of additional autoimmune preclinical models. you don't mind advancing, please. Now, similarly, as we showed you in the last several cases, we can begin to utilize our human biological database to explore the role of this marker in related phenomena, including canonical autoimmunity. Across more than 100,000 individuals, we find that lower levels of this marker are associated with greater risk of IBD, rheumatoid arthritis, and other autoimmune phenomenon as shown in the left panel here. And in the interest of time, as you can imagine, I'm glossing over quite a bit of biology, but we can use our biological database to probe the underlying mechanisms. In this case, identifying the marker is a key indicator of neutrophil activation and tissue-specific honing of neutrophils, which is shown here on the right. Now, interestingly enough, for this particular marker, it's not simply a correlate of autoimmunity. It's also an active driver of underlying disease processes. And repleting this marker back to baseline levels, remember this is a marker that drops in the setting of autoimmune conditions. So repleting back this marker in preclinical models abrogates the underlying autoimmunity observed with immunotherapy without affecting actual tumor efficacy. Now, again, these are just examples that are meant to represent simple conceptual sort of examples of how small molecule biomarkers may be utilized to enrich clinical studies to ultimately make them smaller, faster, and hopefully more effective. If you don't mind advancing, please. Now, I'm happy to go ahead and stop there in the interest of time and answer any questions from the audience and the moderators. Just before we start the Q&A, I would invite anyone on this symposium to visit Sapient on our website at sapient.bio, as shown there on the bottom left. And as you can imagine, here's our contact information, our phone, email, and the Calendly link. And with that, I appreciate your time, and I'm happy to take any questions.
Thank you so much, Dr. Jain. That was really great and very interesting. We do have a number of questions that have come in. Let me start with this one. Dr. Jain, can you describe the process of going from discovery of a small molecule biomarker to actual translation to clinical trials and ultimately practice?
Yeah, it's a great question, Stacy, and this is really the crux of what we do. Discovery, as you can imagine, these markers is only one component, and it doesn't help to discover something quickly if you can't translate it also quickly. And so here at Sapient, we have a high-complexity CLIA laboratory that allows us to not only discover these markers, but to validate them very quickly using more canonical mass spectrometry approaches, and then ultimately advance them then to our CLIA laboratory for development of CLIA-certified assays. And that way we can not only do the R&D, but we can also translate these discoveries to actual clinical tests that can be used as part of both clinical studies or as part of clinical practice.
Great. What is your perspective on the use of singular biomarkers for clinical trial enrichment versus more complex machine learning-based patterning of biomarkers to substratify individuals?
Yeah, it's a good question. And as you saw in the examples that I just showed, there's really no one singular way to approach this. The value of identifying singular markers, of course, is that the translation through CLIA testing and clinical implementation can be quite a bit faster. And obviously there's tremendous value in that. But on the alternative side, we can use much more complex machine learning or AI-based patterning to identify hundreds or even thousands of markers that can begin to provide a much more complex picture of a clinical syndrome, as I showed you in the second example. Now, obviously, there's challenges with translating much more complex patterns through the agency for ultimate approval. But it's something that we do quite routinely to understand underlying biologic phenomenon. So it really relates, Stacy, to the underlying question. If the goal is to identify a singular marker that we can rapidly translate, whether that be a clinical diagnostic marker, a safety marker, et cetera, that is certainly a very valid approach. And alternatively, if the goal is to use much more complex-based machine learning to understand complex disease processes, that's also something that we can do quite readily.
Thank you. Can you please describe Sapiens' underlying human biology database? Is the database representative of healthy individuals or enriched for certain disease types?
Yeah, absolutely. So I realize I glossed over this, but Sapiens' human biological database really consists of hundreds of thousands of biosamples which have been collected from around the world. And these really represent a diverse sort of offerings, if you will, across the disease spectrum. In about half of the individuals, these represent community-based studies in which individuals have biosamples collected under relatively healthy conditions, and then they're followed for 2 to 3 decades of time for the development of disease. And as you can imagine there, you can monitor the transition from a healthy to an abnormal state, as well as understand how early on in the disease process these markers may be altered. And then for the other half of individuals, these come from a number of clinical studies, disease-centered case control studies, et cetera, in which case they're enriched for particular diseases. Now, there's enrichment for certain oncology diseases, you can imagine. There's other studies we have that are enriched in the cardiovascular space, in rare diseases, including pulmonary arterial hypertension or rare genetic diseases. So this really represents a very large, diverse collection of biosamples and secondary databases that have been generated here at Sapiens. And for anyone interested in particular disease areas, I'd be happy to chat further about specific numbers in those areas, but you can think of it as both, Stacy.
Perfect. How do you think clinical trial strategies will continue to evolve over time, particularly in regards to the use of biomarkers for enrichment strategies?
Yeah, this is a good question, and of course I may be somewhat biased in that, but I believe the last couple of years has taught us that there's a need to do this faster, and when it's done faster and with greater efficacy, it can be done in quite a safe way, and really trials have to improve. They have to fundamentally change in a way that allow us to begin to really target those individuals that are having a benefit. It's still remarkable to me, again, going back to some of the data we shared, that over half the individuals that receive a therapy, and that's under a good condition, are really not benefiting much from that therapy. And of course, there's not only a cost to the provider and the patient under those circumstances, but it also means that the patient is not receiving a therapy that would theoretically benefit them, which is really the main cost in my mind. And so I think there really has to be an evolution in clinical trials. Already we've seen this, particularly in the oncology space, where markers and biomarkers have really revolutionized how we think about clinical trials, basket indications, and made clinical trials much more efficacious. And I suspect that advancing forward, this is going to be the norm rather than the exception across a number of different disease areas outside of the oncology space. So this is certainly something that I think is absolutely required, both for those that develop drugs, for those that pay for drugs, and those that sort of utilize the drugs for disease processes. So it's something that's long overdue. I think the FDA recognizes the value of this, as well as those who spend quite a bit of time developing drugs recognize the potential value of identifying these individuals and then these markers that allow us to do this.
Well, this is obviously a very hot topic. We have a lot of questions, so let me, if you don't mind, I'd like to get a few more.
Please.
Can you please provide some thoughts on how biomarkers may be used to substratify individuals with a given diagnosis, and how this information may be used as part of clinical studies?
Yeah, absolutely. And this goes back to the basic question of why only a portion of individuals receiving a therapy actually respond. And that is the way we diagnose disease is really still based upon pathology. And as I showed you in that sort of theoretical example, on pathologic examination, or for any given diagnosis, if you will, individuals can, for all intents and purposes, look uniform, but we know, in fact, that they're not uniform. We know for individuals that have a certain diagnosis, there are those that are going to have a rapid progression and a much greater event rate and a much more sort of adverse course. And there's other individuals that are going to have a much more benign or slow or indolent decline over time. And so these are fundamentally not the same disease, even though they have the same diagnosis or the same pathology. And herein is where I think biomarkers can be extremely valuable in beginning to subset out those individuals who have slightly different biology that drive those disease processes and ultimately allow us to identify those different biological processes and target them in an appropriate manner. And I think fundamentally the way we think about diagnosis as part of these clinical trials has to shift. And already we've seen this again in the oncology space, whereby we think about cancers much more from an oncologic mutation perspective now, as opposed to a particular tissue of origin. And that same concept, I think, is going to port over to many other disease processes. And this is where I think these small molecule biomarkers can be transformative in identifying particular mechanisms by which someone, again, goes from a normal to a disease state, and then ultimately, for a given disease state, how we subset individuals into responders and non-responders based upon biological mechanisms. And already we're beginning to see this when we look at singular diseases, even in the rare genetic space, whereby everyone may have the same exact mutational burden, that there's very different clinical courses, and these small molecule markers can really begin to identify those individuals who are going to sort of have very different clinical courses over time, and ultimately are going to respond very different to therapies.
Great. What is the level of sensitivity of quantification? Can you detect single-digit picograms per ml or lower?
Yeah, it's a complex question, and it has to do with the fact that there's no single answer, because this is not a targeted approach, but rather is a discovery non-targeted approach. The level of quantitation really has to do with the underlying ionization efficiency of a particular molecule, as opposed to a whole as opposed to the underlying LC-MS. And so there are molecules for which the sensitivity can really be quite sort of sensitive in the picograms per ml, or even lower than that. And then there's other molecules, as you can imagine, whose ionization efficiency is not as great, or they're more subject to matrix effects, which makes their sensitivity less pronounced. If across all molecules, you can think of this as somewhere in the low micromolar range is sort of where our median sensitivity is for detection of a molecule. And that's at a 5x signal to noise, which allows for relative quantitation of these molecules.
Great. Let's get another question here. Once you identify a particular single analyte, do you continue to use mass spec to analyze the samples, or do you switch the analysis technology to another platform?
Yeah, so the answer is both, and it really depends upon the specific analyte. Certainly, we develop CLIA-based assays around these single analytes using mass spectrometry all the time, and in most places in the world, doing mass spec-based analytics is quite feasible under a targeted approach, and so we do that quite often. But there are a number of analytes for which we'll also develop point of care or point of contact based testing, and obviously those use alternative technologies, whether they be flow-based technologies, ELISA, et cetera, et cetera. It very much is dependent upon the underlying analyte.
Great. What do you think are the major challenges in biomarker identification and their use in clinical trials?
Yeah, that's a really good question. I think there's a couple of challenges. One has to do with how we sort of fundamentally view and diagnose disease. Again, this has not changed over several thousands of years and is really based upon pathology more than anything else. And obviously we've seen this evolution, again, primarily driven by the oncology space, by which we can begin to sort of define particular disease states based upon markers. So one is a conceptual hurdle in which providers begin to think about diseases in a slightly different way, not one based upon the tissue of origin again, but through biological mechanisms that's largely driven by these markers. The second is really a technical hurdle, Stacy, and that has to do with having underlying technologies that are robust and that are scalable. And the key challenge in this space is these technologies not only have to be robust across small sample sizes, but very large sample sizes on the tens to hundreds of thousands of individuals. And once we can achieve this, this is where we really can sort of enable the discovery process. Again, we've seen this with sequencing and parallel sequencing technologies now that allow us over the last decade or so to really begin to approach these large patient populations, including in the cancer realm, and that's why these markers have come to the forefront. And I think the time is now to think about alternative technologies, and this is where we very much view these types of mass spectrometry technologies that we've developed, in that they can provide orthogonal information. And finally, given the technical advances, are finally scalable to sizes of populations that really enable a robust discovery.
Well, Dr. Jain, that is our last question. Did you have anything you wanted to close us out with?
I really appreciate everyone's time, and Stacy, I appreciate you moderating. For anyone who's interested, as you mentioned, this is a recorded sort of symposium, and we're happy to sort of speak further if anyone has any questions, and certainly don't hesitate to reach out to us on our website, and I appreciate everyone's time again.
Thank you so much, Dr. Jain. Thank you to our participants, and especially thank you to Sapient for partnering with us in this webinar. It was really very informative, and I hope everyone will join us at a future AAPS webinar in the future. Thank you so much, and you can now disconnect from the webinar.